Check it out now, y'all.
NanoHub U, online instruction.
Hello, and welcome back for our fifth and final lecture of NanoHub U's Introduction to Bioelectricity, week 5. So in the preceding weeks, to recapitulate, we began with an introduction to the anatomy and physiology of the nervous system. We continued with an examination of the synapse, the neurotransmitters, and how they give rise to excitatory and inhibitory electrical signals called postsynaptic potentials, how those sum to give action potentials, how we model those action potentials in passive neurons with a cable model, in active neurons or in active processes like an action potential, with a Hodgkin-Huxley model. We talked about Hodgkin-Huxley. We talked about the implications of that model. And we've spent this fifth and final week talking about specific applications of bioelectricity. In this last lecture, I want to switch a little away from electricity, and I want to talk about other ways of getting an electrical response without using an electrical stimulus. So we talked about, when we went through ion channels, we talked about ligand-gated ion channels, we talked about voltage-gated ion channels. So those are ion channels that open and close in response to the presence of a molecule. In the case of a ligand-gated ion channel, that molecule could be a neurotransmitter, a small molecule neurotransmitter, a large molecule peptide neurotransmitter. It could be opening and closing in response to a voltage, like voltage-gated sodium ion channels, for example, at the tissue membrane that lead to the action potential, or voltage-gated calcium ion channels at the terminal of the axon that lead to the influx of calcium, which then leads to the vesicles binding with a presynaptic membrane and releasing neurotransmitter into the synaptic cleft. So there's a number of different kinds of ion channels. In this lecture, we're going to talk about a new kind of ion channel, and these new types of ion channels respond to light, and they are not naturally present in mammals, but we can inject them. We can inject the genetic code for these ion channels into the brain, so that by changing the genetic structure of the neurons, those neurons will then begin to fabricate these ion channels, which will then splice into the neuronal membrane, and that means that we can now open and close these ion channels with light, instead of voltage, instead of ligand-gated ion channels with neurotransmitters, or I should say, in addition to. So it introduces a new way of modulating electrical activity in the brain, and this new way uses light rather than a chemical or a current. And so there's two types of ion channels, but there are many. There's two types that I'll talk about here. One is called channel rhodopsin, and channel rhodopsin is an ion channel that responds to a specific frequency of light, blue light, in such a way that whenever you apply a blue light to it, it opens up, leading to an influx of sodium ions, followed by a depolarizing shift and an action potential with the outflux of potassium ions to restore the resting membrane potential. So in a brain slice, so we can take an animal that has been genetically modified to express these ions, and we can take a brain slice and apply with a laser a blue light, and every time we have a little pulse of blue light, shown here with the blue dot, you can see the electrical signature of the neuron that we've patch clamped. That individual neuron's electrical signature spikes with a blue light. So it's positive confirmation in the laboratory that we are able to modulate that neuron's firing, in a slice at least, using blue light. There's another type of ion channels called halorhodopsins that work in the opposite fashion. They are inhibitory ion channels. And so in this case, applying yellow light with a laser, so a different frequency of light, takes a neuron that has a particular resting firing rate, and when we apply that light, the neuron is silenced. So you can excite a neuron or you can inhibit a neuron. And if you can do either of these two things, then you can begin to ask yourself, what, besides being really cool, what clinical applications or what biomedical applications this sort of technology might have? And the answer is that when you electrically stimulate neurons in the brain, you are not electrically stimulating an individual neuron. That's not how current works. You're pumping current out of an electrode, and the tip of the electrode creates a sphere of charge around that electrode surface. And the intensity, the density of the charge, which leads to the intensity of the potential, drops off with radius squared of that spherical distribution of charge. So you're going to excite a volume of tissue. And you're going to excite a volume of tissue no matter how much you would like to excite an individual neuron. Now, if you get really close to an individual neuron, then you can make your electrode site really, really small and make that excitation so small that the volume only encompasses the adjacent neuron. And that's what biologists mean when they talk about using a high impedance electrode. When biologists say they want a high impedance electrode, they don't actually mean that they want a high impedance electrode. What they mean is they want an electrode with a very small surface area so that they can either stimulate a small volume of cells or they can record from a very small volume of cells. Ideally, what they would like to have is a very small electrode with also a very low impedance, because that impedance, a high impedance, only gives you noise. So really what you want is a small electrode area rather than a high electrode impedance. But when we're looking at that, there are limits to how far we can go. With, with optogenetics, you open up an entirely new set of possibilities in that because it is light rather than electrical charge, which as ions distributes in that fashion in the tissue, it's light, which we can focus on a very, very small spot. So you have a spot of light. Light doesn't diffuse the way that ions do. And so you have a spot of light that allows you to preferentially stimulate or inhibit a very specific electrical circuit within the brain. And so there's that attraction. There's also the possibility that the optogenetic or the genetic modification leading to an animal becoming or a neuron becoming light responsive can be delivered through what's called a viral vector, where the virus, what a virus typically does is it changes the DNA of a neuron or of a cell so that the cell will make more of the virus and then die, and then the virus spreads. But you can make your own virus. You can genetically engineer your own virus so that instead of making more of the virus, it'll make whatever you want it to make. In this case, you could have it make the proteins that make up the ion channels. So you can genetically engineer a virus, you can deliver that virus, and that can be the means that you use to get this optogenetic effect. You can divide the optogenetic effect into two viruses, when one virus will lead to the ion channels and the other virus will lead to the enzyme that splices that ion channel into the neuronal membrane. So you need both viruses to get the excitation from blue light, for example. Now you can inject one virus over here in the brain, and you can inject the other virus over here, and only those cells which go from here to here will receive the genetic information to become optogenetic. So now you can isolate a particular tract in the brain, so a particular projection from one part of the brain to another part of the brain. And when you shine a light on the whole surface of the brain, only those neurons in that projection that you're interested in will be affected. So if you think back to when we talked about, for example, Parkinson's disease, we talked about projections from the subthalamic nucleus to the globus pallidus. And we talked about inhibiting the subthalamic nucleus, and so we stimulate and we affect a volume of tissue. Well, we could just say all we care about is those projections, and then we can preferentially stimulate them. In alcoholism, we talked about projections from the ventral tegmental area to the nucleus accumbens. In epilepsy, we were talking about projections within the hippocampus, from one region of the hippocampus to another region of the hippocampus, setting up a sort of positive feedback loop that leads to this massive synchronized firing that characterizes a seizure. If you can identify the projection of the individual neurons that you're interested in, optogenetics opens up the door to the possibility of delivering a clinical treatment that allows you to preferentially stimulate only those projections you're interested in. Or alternatively, if you're not able to overcome the regulatory barrier that the FDA puts in front of just about any kind of new treatment, you can take optogenetics as a research tool that allows you to understand the projections in the brain, the circuitry in the brain that is associated with epilepsy, and make a determination about which regions of the brain or which tracts and projections in the brain are the ones that are most involved with the rise of seizure phenomena, and then take your electric device and position the electrical stimulating electrodes so that you can take advantage of the knowledge that you gain from optogenetics. So it's an extraordinarily powerful tool, both to understand how the brain works and potentially down the road to alter brain function and show that we have a better understanding and arrive at a better understanding of neuronal circuits. But to do that, you need to move from a preparation in which you're looking at neuronal slices using lasers to deliver light to a preparation in which you can look at a whole animal that is optogenetic animal that has been genetically modified to express these opsins. You can look at the whole animal with a device that is portable so that the animal can move freely and you can do examinations about neural activity in the animal's natural environment. And so that is what we're looking at here. So this is a device developed by a PhD candidate at Purdue named Stephen Lee that incorporates a number of different technologies that we have talked about in the last few lectures into a single device. So you have a single device that has a height of about 1 centimeter, and it has a depth of about 1 centimeter and a width of about 1 centimeter. So it's more or less 1 centimeter cubed. It's a little bit smaller than that. But think of that as a small die, right? So you have a device of about that size, which can be divided up into a series of portions. You have a stack of batteries or supercapacitors. You can use either one. Shown here is thin film lithium batteries, which can be combined with an interface board containing a capacitor to help with the battery. So batteries have very high energy density, but they have very low power density. So they're very good at supplying energy over time, but they're not good at supplying peak currents. That's what power density is for. Capacitors are good at power density, but they're bad at energy density. So by combining a battery with a high energy density with a board that has a capacitor for the high power density, you get the best of both worlds in a combined power supply. That we add to a control plug, which includes a microcontroller and the associated optics, or drivers for the optics that we need, and that can all be plugged into an implanted device. The implanted device now sits inside the cortex, the skull of the brain. It sits inside the cranium in a hole that you dig out, and in that hole you place the ferrule, which contains an optical fiber, and it's coupled surface mounted to the top of the cranium on which the rest of this device sits. And you see all these connectors here because this whole design is modular. It's meant so that you can plug and unplug different parts so you can change what your device does according to the experimental design that you have in mind. So here's a photograph, rather than the cartoon in the previous slide, here's a photograph of the device in question. So you have the implanted optical fiber, and this fiber can have any number of different widths. And so we take this fiber and we push it down into the brain area that we're interested in. Then we lock it in to the cranium with this connector, the LED, which will replace a laser with an LED. So we've gone from a tabletop system that uses watts to a minuscule surface mount system that uses milliwatts, fraction of the total power consumption. And we then have a microcontroller unit on a board that connects to that LED board, and then you have the power board along with the passive radio that allows you to receive programmatic information. So you can tell this device when to stimulate, you can tell it when to stop stimulating. And the first thing we want to do with something like this is begin to characterize it. We know how much light a laser needs in order to excite a neuron that is optogenetic. We need to find out how much light can we generate with an LED. And then we need to make the observation that a neuroscientist working in a laboratory with a laser doesn't particularly care how much light he's using because they have a laser that's plugged into the wall. Power is free. For us, we care very much how much power we're using. And so the neuroscientist using a device like this has to be very conscious of how much power she's using, because power is now expensive. It's contained within a very small battery, and it's resupplied wirelessly, which is a very inefficient link. So we look at that irradiance, the measure of light power produced by a device like this. We look at that irradiance and we plot it as a function of current, and we find that the average device has a linear relationship between current and irradiance shown by the black line, and you have a 95% confidence interval that for any given device you're going to fall somewhere within this gray area. But looking at a large number of devices, we've seen values anywhere from this minimum in this lower dashed line to this maximum in this upper dashed line, and that gives you a sense of the amount of variability from the manufacturer. So we have to take that into account as we select our devices. Once we've done that, we can program two different parameters into the device. We can program the frequency of the stimulus pulses, and we can program the pulse width of these stimulus pulses. So these devices are programmable, and that makes it much easier to adapt the pulsons that we talked about in lecture 5.3 and the automatic neural control algorithm we talked about in lecture 5.2 to implement those algorithms in a device like this by being able to change the waveforms to get the maximal efficacy over time and by able to add the recording functionality that closes that loop for the autonomous neural control. So we have this programming capability. Another thing that we're concerned with is our heating at the surface. So a laser creates an enormous amount of heat. You can have liquid-cooled lasers even. An LED is going to make a lot less heat, but it's also not going to be located on a table. This device is going to be located on the surface of your cranium, just near to your brain. So the question is, how much heat are you creating? And what you're worried about is getting a change in temperature of 1 degree Celsius. So we looked at how much heat are you getting at the device surface as a function of frequency and as a function of current. And we measured a change in temperature when the device is on of about 0.5 degree for peak frequency and about 0.8 of a degree for peak current. So this peak current gives you an irradiance that is higher than we need to stimulate any neuron. So these two together give us a sense that this device is safe, that it's not going to heat the brain. We know that it's small. Can we position the optical fiber in the right part of the brain? And so we designed an experiment in which we would take animals and we would position these optical fibers in that same area of the reward pathway that we looked at in lecture 5.3 and modulate the reward pathways using light. And so we have a target area, the ventral tegmental area, that we wish to position these electrodes in. And so we push these optical electrodes, these fiber optics down, and in a number of different animals, afterwards we do what's called a histology. So after the animal is euthanized, you take the brain, you slice it, and you look at where those electrodes were. And you do that through these tissue slices. And we found all the little red dots are where the electrodes were. So we were in every case where we wanted to be. And then we can look at the slice and see there's the hole that's made in the brain by the electrode itself. There's the tip of the optical fiber, and that's where the light comes out of and you get your stimulation. And then we can zoom in to a portion of the brain and then zoom in further and see different aspects of neuronal density, neurons of interest, neurons that express the opsins that we're after. And so we have confidence that those neurons are optogenetic. And given that confidence, we can then start to look at different populations of animals. So we can look at control animals, and these are animals that are not optogenetic, but have undergone the seizure and have the devices implanted in them. So when you optically stimulate, you would expect to see no change because they're not optogenetic. But this allows us to tease out and make sure that there's nothing about the device or the surgery that's affecting the animal's behavior, and that whatever effect we might see is a result of the light stimulation in the reward pathway. Then we have two populations of animals that we stimulate with different amounts of light to see where that threshold is at which they begin to respond to the optical stimulation. And so over a period of time, you do what's called a conditioned place preference test. So you take a box which has a white region and it has a black region or dark region in it, and you put the animals in it, and mice will preferentially go to the dark region. They prefer the dark to the light. And so you have some distribution of the time that they spend between these two regions. And then what you can do is you can say, you know what, I want you to like the light region better than the dark region. And so what I'm going to do is every time you go into the light region, I'm going to stimulate your reward pathway to make you happy. And every time you go into the dark region, I'm not going to do anything. And so they go back and forth and they get rewarded. And over time, if you are in fact stimulating and you're only shining a light, so this proves, if it works, that these neurons are optogenetic and that we are modifying behavior. Again, if it works, over time you would expect animals to spend more time in the region of the brain, in the region of the cage where they're being stimulated when they're in that part of the cage. And so we can map out the post-test activity for the different populations. And really when it starts to get interesting is when we start looking at preference. So the activity is, are they moving around as much before as they were afterwards? And there's no statistically significant change. So they are in fact maintaining a level of activity. So we're trying to rule out any other variables. That's what the controls are for. That's what this test is for. But let's look at post-test preference. And the question is, what is the preference to the light region over the dark region after they've been rewarded for being in the light region? And what you find is that in the control animals, there's really very little change. They spend most of their time in the dark region. In the lower amplitude stimulation group, there's very little change as well. But in the higher amplitude, you now have a statistically significant change. It's easier to see this data if you plot it just as a delta, as a change in overall. And what you have is the change in preference from pre to post is that the unstimulated animals continue to prefer the dark region of the cage. But the stimulated animal, particularly the one in the second group, has a dramatical change in preference from dark to light. So this animal has effectively become addicted to the optical stimulation, thereby demonstrating that this optogenetic device works in a live behaving animal. And if you look at the dose response curve, you can actually make a map between the amount of light that you're delivering to different populations of animals and the amount of post-test preference that you observe in those animals. And only this last group is statistically significant, so I'll throw that in there as a caution, but leaving statistical significance aside, which we should never do, it does seem to be a trend. Certainly there's a step from no to strong stimulation, where you get a marked change in behavior. We've altered the preference of the animal for one environment to the other using nothing other than light and the genetics of optogenetics. And that's a very interesting achievement. So this is a cutting-edge tool. This is a paper that's just being published this year. It's a cutting-edge tool at the forefront of our examination of bioelectricity. And it introduces the idea of using something other than electricity to modulate electricity in the brain. So that concludes our fifth and final lecture of week 5. I hope you have enjoyed the course, and I look forward to communicating with you directly. Thank you very much.